Yeah, okay. All right. Okay. All right. All right. There you go. All right. So as everyone here knows, obesity is a common, costly, and a serious disease. In United States, the prevalence of obesity is over 40% in adults. Obesity results in over $1.4 trillion in healthcare costs, either direct or indirect. And obesity is associated with a broad spectrum of comorbidities, including cardiovascular disease, high blood pressure, and fatty liver, for example. So obesity and overweight are caused by prolonged periods of positive energy balance. The major contributor to energy expenditure, which promote weight loss, are your basic metabolic processes as well as physical activity. And in addition, thermogenesis mediated by brown adipose tissue also contributes to energy expenditure and promote weight loss. Mammals have three major types of adipocytes, which differ in terms of their functions, morphology, location, as well as developmental origin. The classical brown adipocytes are embryonic origin cell type and are primarily found in the interscapular region. And the beige adipocytes arise in response to a beta adrenergic stimulation or cold exposure within subcutaneous white adipose tissue. And the white adipocyte, the classical white adipocytes, are involved in storing excess calories as triglyceride, and they have a unilocular lipid droplet. In contrast, the brown adipocytes and beige adipocytes have more multilocular lipid droplets. They have lots of mitochondria. And they're involved in promoting energy expenditure through thermogenesis. So brown and beige adipocytes exhibit high expression of a mitochondrial protein called UCP1, which is an inner mitochondrial membrane protein which uncouples respiration from ATP synthesis, allows protons to leak across the inner mitochondrial membrane and bypass ATP synthase. This results in generation of heat and increases energy expenditure. In addition, so the UCP1 is a classical mediator of non-shivering thermogenesis. In addition to the UCP1 pathway, a couple of other UCP1 independent pathways have been identified. This includes futile calcium cycle mediated by Circa family of calcium pumps. In addition, another pathway involving creatine cycle is involved in brown and beige fat thermogenesis. And in addition to these mechanisms of thermogenesis, previous studies suggest that mitochondrial morphology is also an important regulator of brown fat mediated thermogenesis. Work specifically from Orion Sherihai lab suggests that norepinephrine stimulated mitochondrial fission acts as an amplification signal for thermogenesis, and it involves phosphorylation of a fission factor called GRP1 which gets recruited into mitochondria and promotes mitochondrial fission. All right, so mitochondria are highly dynamic organelle that modify their abundant and morphology depending on the metabolic context. Usually in starvation condition where there's increased need for ATP synthesis, you tend to see more elongated mitochondria. In contrast, when there is nutrient surplus, you tend to see more fragmented mitochondria. And so fragmented mitochondria are more associated with decreased energy efficiency, bioenergetic efficiency. And essentially, that's what brown adipocytes exhibit, a decrease in bioenergetic efficiency and more uncoupled respiration. So when you think of mitochondrial fragmentation, people generally think it's deleterious. For example, in cardiomyocytes, increased mitochondrial fission is deleterious. However, in brown adipocyte, it's not necessarily a bad thing because by function, brown adipocytes are involved in uncoupled respiration, and mitochondrial fission promotes that. All right, so mitochondrial fusion, this is just a basic diagram, just basically involves fusion of two mitochondrion into one, and fission involves splitting of one mitochondrion into two. So mitochondrial fusion is mediated, outer mitochondrial, of the outer mitochondrial membrane is mediated by the mitofusin 1 and mitofusin 2. The inner mitochondrial membrane fusion is mediated by a protein called OPA1. And fission is initiated when endoplasmic reticulum gets recruited to the site of mitochondrial fission, which is marked by mitochondrial DNA. So basically, the ER wraps around mitochondria and promotes mitochondrial fission. This subsequently results in recruitment of a fission factor called GRP1, which aids in the ER-mediated constriction of the mitochondrial membrane. All right, so like mitochondria, peroxisomes are also highly dynamic organelles. And this is what my lab focus on, the role of peroxisomes. So peroxisomes are multifunctional organelles that are involved in beta oxidation of very long chain fatty acid, 22 carbon or longer, which cannot be oxidized in mitochondria. In addition, they're involved in beta oxidation and alpha oxidation of methyl branched fatty acids, as well as synthesis of bile acid and a special class of phospholipid called ether lipids, which are phospholipid that have an ether bond at the SN1 position of their glycerol backbone. And the most common form of ether lipid are called plasmalogens. All right, so work from my lab, as well as others, suggests that peroxisomes play an important role in metabolic homeostasis. Recent studies suggest that certain species of these ether lipid called plasmalogens are significantly decreased in obese individuals as opposed to lean individuals. Studies from our lab, as well as others, suggest that peroxisomal beta oxidations affects lipid homeostasis in the liver through multiple mechanisms, including inhibition of lipid hydrolysis through lipophagy or lipolysis, reactive oxygen species from increased beta oxidations have been implicated in lipotoxicity in beta cells, which impairs glucose-stimulated insulin secretion, increased oxidative stress from peroxisomal as well as mitochondrial fatty acid oxidation also contributes to insulin resistance in adipose tissue. And our work suggests that peroxisomal plasmalogens are important for regulating mitochondrial fission and promote energy expenditure. So this is what my talk today will focus on. All right, so peroxisomes, de novo formation of peroxisomes involves fusion of a pre-peroxisomal vesicle from the ER with a pre-peroxisomal vesicle derived from mitochondria. And this is followed by recruitment of peroxisomal membrane as well as peroxisomal matrix proteins. And this process involves a dozen or so protein called peroxins or PEX proteins, which affect different aspects of peroxisomal biogenesis, including factors that are involved in assembly of peroxisomal membrane, factors that act as chaperones for peroxisomal matrix proteins. The peroxisomal matrix proteins are synthesized in the cytosol and have to be imported into peroxisomes, and there are chaperones that involved in that function. In addition, there are PEX proteins that act as docking receptor for these chaperones and recycling them back into the cytosol. So three peroxisomes in particular are very important for peroxisomal formation, de novo formation of peroxisome. These include PEX3, PEX16, and PEX19. Loss of function mutations in these peroxisomes results in complete ablation of peroxisomes and are associated with devastating genetic disorders called the Zellweger syndrome. All right, so my lab is interested in the role of peroxisomes in adipose tissue. So we wanted to know what happens to peroxisomes with high-fat feeding. So we noticed that when we feed the mice a high-fat diet, the peroxisomal biogenesis genes significantly increase in brown adipose tissue, both at the gene expression level as well as the protein level. We also wanted to know what happens in response to cold exposure, and noticed that both in the classical brown adipose tissue as well as in the subcutaneous white adipose tissue, which has increased propensity to turn into beige fat, there is increased expression of peroxisomal protein with cold exposure, suggesting that peroxisomes may be involved in thermogenesis. All right, so thermogenic gene expression involves requires this very large transcription factor called PRDM16, which is a transcriptional coactivator that interacts with other transcriptional factors such as PPAR gamma and EBF2 to drive gene expression in classical brown adipocyte as well as in beige adipocytes. And so we wanted to know whether PRDM16 is important for cold-induced peroxisome biogenesis. To address this question, we generated mice with adipose-specific knockout of PRDM16, and then either kept them at room temperature or subjected them to cold exposure. And as you can see here, PRDM16 is not important for constitutive peroxisome biogenesis. However, it is definitely required for cold-induced peroxisome biogenesis, suggesting that transcriptional regulation of thermogenesis as well as de novo peroxisome formation are coordinated. All right, so we wanted to know what is the role of peroxisomes in adipose tissue, and decided to knock out PEX16. So I mentioned that PEX16 is one of the peroxin that's absolutely required for peroxisome biogenesis. So we generated mice with adipose-specific knockout of PEX16, and this results in degradation of other peroxisomal proteins. So there's no, I didn't show here, but there's no effect on gene expression of PMP70 or ACOX1, but the protein levels are dramatically decreased, presumably because in the absence of peroxisome, so you don't see any peroxisome in the PEX16 knockout, these peroxisomal proteins get degraded. All right, so these mice on chow diet had essentially no phenotype. They look perfectly healthy. However, when we fed these mice a high-fat diet, they weigh significantly more, have increased fat mass. The gonadal white adipose tissue weighed significantly more. The adipocytes tend to be larger in the knockout. We also did histologic analysis of the classical brown fat and noticed that there is increased accumulation of lipids, triglycerides, suggesting that they have impaired thermogenesis. All right, and so we wanted to look at if the thermogenesis is affected. Yeah, dramatically. These mice are not able to tolerate cold exposure, and in fact, they die if you keep them in cold. We also looked at beta agonist stimulated energy expenditure, which is a proxy for thermogenesis activation, and it's significantly decreased in the PEX16 knockout. This story was published in JCI a few years ago. I won't show you the mechanistic data because I want to show you unpublished data, but I'll briefly summarize the mechanistic data from this paper. So basically, our data suggests that cold results in activation of PRDM16. PRDM16 in turn derives peroxisome biogenesis, and peroxisomes are involved in synthesis of ether lipids called plasmalogens. And we showed in this paper that plasmalogens are a component of the mitochondrial membrane and are important for mitochondrial fission. So, we showed that these mice, when we knock out PEX16, they are more obese and have impaired mitochondrial fission. And interestingly, if we give back plasmalogens, we can rescue this phenotype. All right, so now I'll show you our unpublished data that stems from this work. So a very talented postdoc in my lab, Donghua Hu, was interested in better understanding the molecular mechanism through which peroxisomes affect mitochondrial dynamics. So first, he wanted to validate that peroxisome deficiency results in effect on mitochondrial fission. So he knocked out PEX16 in brown adipocytes, and as you can see here, as compared to control adipocytes, they have much more elongated mitochondria. And as a result, since mitochondrial fission and mitochondrial DNA replication are coupled, the mitochondrial DNA content is significantly decreased, suggesting that peroxisome deficiency results in impaired mitochondrial fission. So our results also suggest that that plasmalogens are important for the role of peroxisomes in mitochondrial dynamics. So this is the peroxisomal plasmalogen synthetic pathway. Basically, the synthetic pathway, the peroxisomes are involved in the initial steps of this pathway and generate a metabolite called alkylglycerol, which then moves to the ER and undergoes further metabolism and results in production of these lipids called plasmalogen. And this is a structure of that. Basically, these are glycerophospholipid, and they have a fatty acyl chain at the SN2 position. At the SN1 position, there's an alkyl chain. There's a loss of the carbonyl oxygen. And these lipids are important for thermogenesis. So when we knock down GNPET, which catalyzes the rate limiting step in plasmalogen synthesis, you get a phenotype that resembles the PEX16 knockout. You get more elongated mitochondria. And interestingly, if we give back this precursor, which enters the synthetic pathway downstream of the peroxisomal step, it essentially completely rescues mitochondrial fission, suggesting that peroxisome-derived lipids regulate mitochondrial fission. And so we can rescue the mitoDNA content by giving back plasmalogens. All right, so we wanted to understand the molecular mechanism through which peroxisomes regulate mitochondrial fission. So we decided to isolate mitochondria from PEX16 knockout mice or the wild type mice that had been subjected to cold exposure, and did untargeted proteomics analysis. And many proteins were either increased or decreased in mitochondria of these peroxisome deficient brown adipose tissue. And so the protein that increased include PEX14 and this protein called PHYH, which is involved in alpha oxidation in peroxisome. So this suggests that certain peroxisomal proteins in the absence of peroxisomes are mistargeted to mitochondria, and other proteins are decreased in mitochondria. In particular, a protein called TMEM135 was the most decreased protein in mitochondria of peroxisome deficient brown adipose tissue. So this decreased level of TMEM135 in brown fat was not because its gene expression was decreased, and it's not because the protein is degraded, because when we look at whole tissue lysate, there's no difference in the level of TMEM135 in brown adipose tissue. So it's in the absence of peroxisome, this protein it no longer localizes to mitochondria. So TMEM135 really piqued our interest because Dr. Ikeda's lab beautifully showed that TMEM135 is an important regulator of mitochondrial fission. So we got really excited that this could be the mediator of peroxisomal regulation of mitochondrial dynamics. So this protein still remains somewhat poorly. We don't really know what exactly this protein does. So it's a transmembrane protein, as the name suggests. It has a 5 transmembrane domain. It has an N-terminal cysteine-rich region. It has a C-terminal tin-17 domain. And in addition, it has a putative targeting sequence for PEX19. So PEX19 is a chaperone for peroxisomal membrane protein. And so we wanted to know whether TMEM135 interacts with PEX19. And so using co-immunoprecipitation, we show that indeed TMEM135 does interact with PEX19. All right, so we wanted to know where exactly is this protein located, just in wild type brown adipocytes. So as expected, it's in peroxisomes, as well as in mitochondria, but surprisingly also in the nucleus. When we ablate peroxisomes in brown adipocytes, it's consistent with our proteomics data. Its level in mitochondria are significantly decreased, suggesting that peroxisomes are required for mitochondrial localization of this protein. So I mentioned that peroxisome-derived lipids are important for regulation of mitochondrial dynamics. So next we ask the question, are these lipids important for mitochondrial localization of TMEM135? So using confocal microscopy, we show that in control brown adipocytes, TMEM135 is strongly localized in mitochondria. However, when we knock down the plasmalogen synthetic protein, now TMEM135 is mislocalized to the nucleus. And interestingly, if we give back the precursor for plasmalogen, we see more fragmented mitochondria as compared to here, and now TMEM is more localized in mitochondria, suggesting that peroxisome-derived lipids are required for mitochondrial localization of TMEM135. All right, so we wanted to characterize this protein. First looking at its tissue distribution. So it's expressed in multiple tissues, but it's more enriched in brown adipose tissue, and its expression significantly increases with cold exposure. And Western blot analysis shows a dramatic increase in the TMEM protein levels in brown fat with cold exposure. In contrast, if we put the mice at thermal neutrality, now the TMEM expression significantly decreased, suggesting that the expression of this protein is regulated by thermogenic stimuli. All right, so we wanted to know, what is it doing? Is it affecting mitochondrial dynamics in brown adipocytes? So we knocked it down in brown adipocytes, and consistent, similar to what we see with the PEX16 knockout cells, you get more elongated mitochondria when TMEM135 is knocked down. The mitochondrial DNA content is significantly decreased, and this is not because of an effect on gene expression of factors involved in mitochondrial dynamics. And consistent with the impaired mitochondrial fission and decreased mitochondrial DNA, which disrupts the oxidative phosphorylation, we see significantly decreased OCR when TMEM135 is knocked down. So encouraged by these in vitro studies, we decided to generate TMEM135 conditional knockout mice. So using CRISPR, we generated TMEM135 flux mice and then crossed them with adiponectin-3 to knock it out in adipose tissue. So this is just a Western blot showing that its expression is decreased in brown adipose tissue. So first, we did ultrastructural analysis to look at what happens to mitochondria. So similar to what happens in the PEX16 knockout, and what happens in the in vitro knockdown of TMEM135, you get more elongated mitochondria in the PEX16 adipose-specific knockout brown adipose tissue. This is just quantified here, the aspect ratio, and the number of mitochondria is significantly decreased. All right, so next we wanted to know, how is this affecting thermogenesis? So we put we subjected the mice to cold exposure, and similar to the PEC16 knockout mice, these mice are less cold tolerant. And we also looked at their oxygen consumption in response to beta agonists, and it's significantly lower, suggesting that these they have impaired thermogenesis when mitochondrial fission is impaired. All right, so next we did some metabolic phenotyping, looking at the body weight. These mice weigh significantly more as compared to wild-type animals when fed a high-fat diet. So there's no phenotype on chow diet, but when we feed the mice high-fat diet, the knockout animals weigh significantly more. It's not because of a difference in food intake. And also we looked at systemic glucose metabolism. As you can see here, the mice are glucose intolerant and are hyperinsulinemic, suggesting that they have impaired insulin sensitivity, and insulin tolerance testing support that these mice are obese and are insulin resistant. All right, so next we wanted to know what happens if you overexpress TMEM135. So when we overexpress TMEM135 in brown adipocytes, now you get more fragmented mitochondria, and mitochondrial DNA content is increased, and it's not because of an effect on mitochondrial dynamic factors involved in mitochondrial dynamics. So this suggests that TMEM135 is not only necessary, but is also sufficient for mitochondrial fission. All right, so in collaboration with Aki's lab, we also wanted to see what happens when we overexpress this protein in mice. And so this is just gene expression showing that TMEM expression is significantly increased in different adipose depots of the transgenic animals. And so when we feed these mice normal chow diet, there's no effect on body weight. However, when we feed these mice a high-fat diet, the transgenic animals weigh significantly less than the wild-type animals. So when you knock out TMEM135, the mice are more obese. However, when you overexpress it, they weigh less. And it's not because they eat less or they have malabsorption. So it's because of increased energy expenditure. All right, so we also did body composition analysis, and they have significantly less fat mass. Histologic analysis shows profound protection against high-fat induced browning of the whitening of the brown fat. So as you can see here, the wild-type mice, you get this noticeable whitening of the brown fat, and this is completely protected in the transgenic animal. The white adipocytes in the inguinal depot are markedly smaller. The liver has a dramatic effect. When you feed the mice high-fat diet, you get this massive fatty liver. However, in the transgenic animal, they're completely protected from fatty liver. As a result, there is a dramatic effect on glucose homeostasis, as you can see here. And the mice are hypoinsulinemic, suggesting that they're insulin sensitive. And yeah, that's what the insulin tolerance testing suggests. So when you overexpress TMEM135, the mice are protected against high-fat diet-induced obesity and protected against fatty liver and insulin resistance. And this is another way to show that is with a AKT phosphorylation, which is markedly increased in the transgenic animals. So next, we wanted to know if they're more cold tolerant. So to the knockout animals, I showed that are less cold tolerant. So we wanted to know if the transgenic are more cold tolerant, and yes. So the wild type mice, so the way we did this experiment is we first kept these mice at thermal neutrality and then subjected them to cold exposure. So the wild type mice are, they're under these conditions, when there's like whitening of the brown fat, they're not able to tolerate cold exposure. However, the transgenic animals are perfectly happy. They're able to maintain their temperature. And this is not because of increased expression of UCP1 or other thermogenic genes or effect on mitochondrial fission factors. So next we wanted to know whether TMM135 is the mediator of peroxisomal regulation of mitochondrial dynamics. So I showed you that this protein was dramatically decreased in the peroxisome deficient brown adipose tissue. So we wanted to know whether if you overexpress it, can it rescue the phenotype in the PEX16 knockout? So we crossed the TMM135 transgenic animals with the PEX16 knockout animals, and as you can see here, that PEX16 is knocked out, and then when you overexpress TMM135, expression is increased. So we did ultrastructural analysis. So consistent with our published data, when PEX16 is knocked out, you get more elongated mitochondria. However, if you overexpress TMM135 on this background, now you get more circular fragmented mitochondria. And the aspect ratio is quantified here. We also looked at the mitoDNA, and overexpression of TMM135 increases the mitoDNA in the PEX16 knockout brown adipose tissue. So we also wanted to know, does it rescue thermogenesis? And we put the mice in the cold. So the PEX16 knockout animals are in green, and they're not able to tolerate cold. However, if we overexpress TMM135, now they're able to tolerate cold, suggesting that TMM135 is likely the primary mediator of peroxisomal regulation of mitochondrial dynamics. So I showed this earlier. Mitochondrial fission involves recruitment of ER to the site of mitochondrial fission. So we wanted to know whether peroxisomes are also recruited to mitochondria during mitochondrial fission. So to do this, we took mice and either kept them at room temperature or subjected them to cold exposure, and then stained the brown adipose tissue either with the peroxisome marker, PMP70, or the mitochondrial marker. And as you can see here, when we put the mice in cold, there is more co-association between peroxisomes and mitochondria, suggesting that there is a potential recruitment of peroxisomes to mitochondria. We also show using norepinephrine treatment in brown adipocyte, there is increased association between peroxisomes and mitochondria. All right, so next we wanted to know whether TMM itself moves from peroxisomes to mitochondria in response to a thermogenic stimuli. All right, so to do this, we took brown adipocytes and expressed lentivirus for peroxisome marker, GFP tag PTS1, or the blue fluorescent tagged TMM135, and then stain the brown adipocytes with TMRE to mark the mitochondrial network. So as you can see here, when we treated the adipocytes with norepinephrine, there is increased localization of TMEM135 with the mitochondrial marker, suggesting that thermogenic stimulus such as norepinephrine promotes recruitment of TMEM135 to mitochondria. So it appears to move from peroxisome to mitochondria in response to a thermogenic stimulus. So next, we wanted to know, does this protein affect phosphorylation of GRP1? So mitochondrial fission involves phosphorylation and mitochondrial recruitment of GRP1. So what happens to GRP phosphorylation when we ablate TMEM135? So as you can see here, it is markedly inhibited, GRP1 phosphorylation. In contrast, when we overexpress TMEM135, now you see more phosphorylation of the fission factor. So this phosphorylation event in brown adipocytes promotes recruitment of GRP to the mitochondria. So we wanted to know what happens to the GRP localization when we overexpress TMEM135. So now, when we overexpress TMEM135, there's more mitochondrial localization of GRP1. All right, so how exactly is TMEM135 doing this? So we search for its interacting protein, and it turns out that TMEM135 interacts with a number of peroxisomal proteins such as PEX3, ACBD5, and PXMP2. In addition, it interacts with the protein called ACAP1. So I showed in the beginning of the talk that the GRP phosphorylation at the serine 600 is mediated by pKa. So ACAP1 is an anchoring protein for pKa. So it appears that TMEM135 interacts with this anchoring protein for pKa and mediates phosphorylation of GRP1. So first we wanted to validate this interaction, and using GST pull-down assay, we show that TMEM135 does interact with ACAP1. We further confirmed this interaction by immunofluorescence analysis. All right, so next, so this suggests that GRP1 interacts with this protein, which is an anchoring protein for pKa, and is localized on the outer mitochondrial membrane, and the complex in turn is required for recruitment of GRP to the mitochondrial membrane. So next, we wanted to study the translational potential of TM135. So consistent with what we see in the mouse brown adipocytes, in human brown adipocyte, when we treat the cells with the forskolin, there's increased expression of TMEM135. When we also wanted to know what happens when we knock out TMEM135. So we're using CRISPR, we knocked it out, and similar to what we see in the mouse adipocyte, you have more tubular mitochondria in the human brown adipocytes. And mitochondrial DNA is significantly decreased, and mitochondrial respiration is impaired. All right, so next, we searched the database, and turns out that TMEM135 expression in adipose tissue is significantly decreased in obese individuals as compared to lean individuals. And in addition, we found that there is a missense mutation that's associated with increased BMI in Hispanic population. So this is a G369E glycine to glutamic acid mutation in a highly conserved region after the TIM17 domain. Okay, so to see if how this mutation affects mitochondrial dynamics, we mutated this site and expressed it in brown adipocytes. So here's the TMEM135 knockout human adipocytes, and in the middle one we re-express the wild type TMEM135 or the mutant TMEM135. So as you can see here, the expression of the wild type and mutant is similar. When we looked at the mitochondrial morphology in these human brown adipocytes, so again in the knockout brown adipocyte, you get more tubular mitochondria. When we re-express the wild type adipocyte, it rescues mitochondrial fission, so you get more fragmented mitochondria. However, when we re-express this mutant associated with increased BMI, now it essentially resembles the knockout. So suggesting that TMEM135 might be relevant to metabolic homeostasis in human through its role in mitochondrial fission. All right, so just to summarize this story, so our data suggests that peroxisome are recruited to mitochondria in response to thermogenic stimuli, and peroxisomes cooperate with endoplasmic reticulum to synthesize these lipids called plasmalogens, which are incorporated into mitochondrial membrane, presumably the outer mitochondrial membrane, and they're involved in fusion of the vesicle, these peroxisomes with the mitochondrial outer membrane, and this in turn is important for translocation of TMEM from peroxisomes to mitochondria. So once TMEM135 is in mitochondria, it interacts with ACAP1 and in turn promotes phosphorylation of DRP1, resulting in activation of mitochondrial fission, and this is associated with increased thermogenesis and energy expenditure. All right, so with that, I would like to thank members of my lab, in particular Danghua, postdoc in the lab who did this work, as well as other members of the lab, and our collaborators, in particular Aki for his generous help in sharing the mice with us and helpful discussion. And my lab is funded by NIDDK as well as these other sources, and I would be happy to take any questions. Yeah, Andrea.
Beautiful work.
Thank you.
There is one thing that I may have missed, and is the rescue of the TMEM135 of the phenol of the PEC16 knockout.
Yeah.
So you showed at the beginning that the PEC16 they get to have no peroxide.
Yes.
And in your model, TMEM135 was downstream of the peroxide.
Right, right.
So how can you rescue the phenotype
right.
if you're missing basically that?
Yeah, so if you overexpress TMEM135, it goes to mitochondria. So when I proposed this, in the grant, I didn't think it was going to work. I thought we were going to have to treat the mice with plasmalogens in addition to overexpression, but it turns out when you overexpress it, it just goes to mitochondria, and it beautifully rescues.
Plasmalogen is not required.
It's not required, but especially in the context of overexpression, which is non-physiologic. So plasmalogens are permissive for its recruitment to mitochondria. So under normal circumstances, I think it is definitely important, but when you overexpress it, it does not appear to be required.
So do you think that?
And also, just because you don't have peroxisome, you can get plasmalogens from the circulation, from other tissues that are imported by brown adipose tissue, and they might be helping in the mitochondrial localization of TMEM135.
So you have not rescued the peroxisomal phenotype with the overexpression. So at some point, I was thinking that the overexpression might substitute for PEC16 function within the peroxisome. And we looked at this in the knockout and genic whether you have more peroxisome.
Well, so if on the PEC16 knockout, you won't have any peroxisome. So there's
and you still don't have them if you overexpress them.
Correct. Yeah. PEC16 is definitely required for peroxisome biogenesis. So even if you overexpress TMEM, you still won't have any peroxisomes. Yeah.
I'm curious what you think TMEM is doing in the nucleus.
Yeah, that's a great question. Aki and I were talking about it. I mean, I don't know. It might be involved in mitonuclear signaling, or it could have some transcriptional role. But at some point, I think we're going to look into that. But it's really interesting that we have this transmembrane protein. How is it going to the nucleus? I don't really know. But there's precedent for that. There's a protein called tetraspanin 8, I believe. It's a 4-pass transmembrane protein that translocates to nucleus. So somehow TMEM is going to the nucleus, and what it's doing, I don't know.
Just to follow up to that, I'm curious, then do you think plasmalogens are not like a major sort of contributor to this localization? Because under those conditions, when you knock down the rate limiting enzyme, you see really strong localizations in the nucleus. So
yeah, so it appears that plasmalogens are not important for its nuclear localization, but it is appears to be involved in its mitochondrial localization. Yeah, so I think that plasmalogens are mediating fusion between peroxisome membrane and mitochondrial membrane. There's some evidence that suggests that that's what peroxisomes do. They're involved in fusion events. Membrane fusion events, because they lack the carbonyl oxygen. There's more interaction between the head groups in phospholipid, and there's more tighter packing of the phospholipid, and it affects membrane dynamics.
So it's interesting when you did your proteomic study where you found that increased in mitochondria, but you saw increased PEC14 on the mitochondria.
Yeah.
Do you start to see accumulation of peroxisomal enzymes within the mitochondria?
Yeah, so PEX14 is not the only one. We noticed that a lot of proteins or several proteins involved in alpha oxidation also move to peroxisome, also move to mitochondria. So, I mean, there's a strong link, metabolic link between peroxisomes and mitochondria. They tend to influence each other's function. And certain peroxisomal protein get degraded, as I showed, in the Western blot, such as like PMP70, it gets degraded. ACOX-1 gets degraded, but then other peroxisomal protein get mistargeted to mitochondria.
That's a really interesting finding. Surface peroxisomal functions of the brain.
Yeah.
In the absence of functional peroxisome.
Right.
That would be really
Yeah. Yeah, actually, the reviewers asked us to validate that result, and we showed. I didn't show it here, but we did immunofluorescence analysis, and there's definitely more colocalization of PEX14 in mitochondria in the knockout cells. When we overexpress it, yeah.
Yeah, I wonder if that's the or if there's
Yeah, that's a really good question. There's a strong link between mitochondrial DNA replication and mitochondrial fission. I think it's necessary to ensure that the daughter mitochondria get equal complement of DNA. So when you promote mitochondrial fission, it also promotes mitochondrial replication, I think, the DNA replication. Yeah.
Regarding the difference between small and high,
Yeah.
was the adult.
Yeah.
So can you be sure that the difference is seen or because of the work?
Well, the mice, when you feed them high-fat diet, they tend to eat a little bit less because there's more. Like when you feed the mice chow diet, I think they eat usually like 4 grams per day, but then when you put them on high-fat diet, they usually eat like 2.5 grams a day because they're getting more. It's more caloric. But over time, they gain weight on the high-fat diet. I don't think that this phenotype is related to the food intake. We measured the food intake prior to difference in body weight, and there was no difference in how much they eat.
Can we go back to the peroxisomal? I was kind of amazed that your mice were fine. You said that there was no effect of the peroxisomal deficiency. Is that what you expected, or other phenotypes of the problem? Brain function, or that you
in our mice, so this is. Are you talking about the? Oh, the PEX16 knockout. Yeah, this was an adipose-specific knockout. Yeah, we didn't look at the brain. But I'm getting... Excuse me? No, their peroxisomes are essentially everywhere in all cells. They tend to be more abundant in brown adipocytes and increase with thermogenic stimuli, but they're everywhere. And in brain, I'm sure that they play an important role, which has not been studied. Peroxisome-derived lipids, plasmalogens, are abundantly expressed in brain. So I'm pretty sure that if you knock it out in brain, it's going to have a severe effect. And Zellweger syndrome patients have severe neurological deficits. Yeah. The transgenic animals were leaner, whereas the knockout were more obese.
But not unless they were fed.
Yeah. So thermogenesis, can be activated by cold or by diet. So when you feed Western diet, there's more diet-induced thermogenesis, and that might be impaired in the knockout. Any other questions? No? Yeah.
And the effects of the one.
135.
You showed that they recovered they that's the relatively better than the other licensing.
Yeah.
Is there a clear connection between, it's really the only like molecular thing that you that was mitochondrial fusion? The increase in cell fat content and increase in thermogenesis, does that have like clear physiological connection to efficiency, or are there potentially other ways in which is regulating those?
Yeah. So how mitochondrial dynamics affect uncoupled respiration? It's not really clear. But definitely, when you promote mitochondrial fission, it's thought to act as an amplification signal. So there's more mitochondria, and there's evidence that round, circular fragmented mitochondria tend to exhibit more uncoupled respiration. It's not really clear why, but definitely the mitochondrial morphology affects uncoupled respiration. Yeah.
What would be a couple of long-term mitochondrial dysfunction, right? If you look at this from the therapeutic,
Yeah.
certain species of vector or this use of mitochondrial. A certain time period probably
Right.
long term symptoms of that can do that essentially.
Right. Yeah. In brown adipocytes, we don't see deleterious effect. But I can see that, hyperactivation of mitochondrial fission could have deleterious effect. And it's been shown in multiple other cell types, especially like cardiomyocytes and beta cells, increased mitochondrial fission is bad. But in brown adipocytes, I think they seem to be perfectly fine because, mitochondrial fission, as I mentioned, is associated with more uncoupled respiration, and that's what brown adipocytes are designed to do. But I don't know, if we, have the mice for a long time, maybe what happens to them. Yeah.
We have a question that was really good from Gisela, saying, asking if are the plasmogens
Plasmalogens
embedded in the mitochondrial membrane?
Yeah, so we don't know if it's outer membrane or inner, but most likely it's outer. So we isolated mitochondria and did lipidomic analysis, and we see plasmalogens in mitochondria, and if PEC16 is knocked out, then there's decreased plasmalogens. Yeah, so plasmalogens are, I showed that peroxisomes are involved in initial steps of plasmalogen synthesis, and the completion occurs in the ER. So it makes sense that the ER is recruited to the mitochondria and peroxisomes are recruited to mitochondria. So yeah, so it's probably going from the ER to mitochondria.
Thank you so much.
Thank you.